So chapter 16 is called Host Microbe Interactions. The first thing that I want you to remember is that most microbes are harmless. Okay, so we have the microbiome that's basically part of us, and those organisms don't hurt us unless something goes wrong. A lot of microorganisms are beneficial, and if you think about the environment, there's a lot of organisms that are beneficial. Okay, now some microorganisms are called, we say they're opportunistic. So they're going to cause disease if there's an opportunity to cause a disease. So an example of what provides an opportunity is someone that might be immunocompromised. So those are people that have something going on with their immune system, and some part of their innate or their adaptive immune system isn't working properly, and therefore the pathogens can sneak past them. Okay, so things that can lead to being immunocompromised, so malnutrition, cancer treatments, right? AIDS, if someone has a surgery or they have a wound, right? A wound provides an opening for organisms to get in. Something that's genetically wrong, alcohol or drug abuse can cause problem, or if immunosuppressive therapy, so if someone has had an organ transplant and they put them on immune suppressors, it could cause a problem. They're more susceptible to microbes that are normally harmless. Okay, so the anatomical barriers as ecosystems. Okay, so there's a bunch of vocabulary words that I want to go over with you. So symbiosis is when two organisms live together. Okay, so you have this symbiotic relationship. So the two of you are together. Okay, now there's subcategories within symbiosis. Okay, so there's mutualism, which means both partners are going to benefit. So it's good for both groups. So for example, in our large intestines, we have bacteria, and the bacteria actually synthesize vitamin K for us, but then we supply the habitat, the nutrients that they need in order to live. So both partners are benefiting from that. Commensalism is one partner benefits and the other is unharmed. So that one I think is kind of hard to prove, because how do you know that the other one is unharmed if it's a microorganism? But so lots of organisms live on our skin. They don't do anything. They consider them neutral, but they get food from the host. Okay, and then parasitism is one organism benefits at the expense of the other. So I always think blood-sucking parasite. Okay, so the parasite is benefiting, but whoever's getting their blood sucked is not. So like fleas and ticks, lice, stuff like that. Technically, all pathogens are parasites, but they use parasites specifically for eukaryotes, so protozoa or helminths, which are worms, or they could use it for insects. Okay, now more vocabulary. Okay, so colonization, that just means that the microorganism is living on the surface. So it could be living on your skin. Okay, so it's hanging out, no big deal. You're colonized, or the plate has a colony, right? So now infection is used if it's a pathogen. Okay, so if it's going to start causing a problem. Okay, so if it's subclinical, that means that there's no symptoms, or if you have symptoms, they're mild. Okay, now what's a symptom? So a symptom is what the patient experiences. Okay, so they're going to come in and they're going to say, oh, I have a headache, I have a sore throat. But the signs are what the healthcare provider can actually observe. Okay, now the initial infection, the first time you get something, that is considered a primary infection. Okay, now a primary infection can predispose people to a secondary infection. So if you think about it, you get a cold, okay, and your nose is runny and you're all full of phlegm, mucus. Okay, that provides a really awesome place for, so it starts out as a virus. So that provides a really awesome place for bacteria to grow. So then the bacteria might end up giving you an ear infection or a sinus infection. Okay, so the primary infection was the cold, but then the secondary infection is like the sinus infection. Okay, a primary pathogen causes disease in somebody that's healthy. An opportunistic pathogen, which I just said, is opportunistic, right? So it's going to infect somebody that is immunocompromised or where there is some sort of problem with, like maybe there's a wound. Okay, so the organism could be there, so it's present, but it takes advantage of the situation and then it gets inside. And I don't know why I put location. I don't know what I was thinking. Okay, now virulence is how pathogenic something is. So is it really bad? Is it super virulent, or is it not very virulent? And so how bad is the disease that it causes? Okay, virulence factors are things that the organism has, or like genes that it has that allows it to make a protein. And virulence factors are what's going to allow the microorganism to cause the disease. Okay, now characteristics of infectious disease. So it can be communicable or contagious, and those are basically the same thing. It can spread, right? It can go from one person to another. Okay, now the infectious dose is the number of microbes that have to get into the host in order to establish infection. Okay, so there's a very wide range of infectious doses. Okay, so Shigella, which this causes diarrhea, right? This is, you ingest it. It only takes 10 to 100 bacteria to cause shigellosis, right? So not very many at all. Okay, but Salmonella, this is 10 to the 6th, right? So 10, 1, 2, 3, 4, 6, right? So 1 million. So 1 million bacteria are required to make you sick from Salmonella. Okay, so the difference with these two, because they're both GI things, it's if the organisms can survive the stomach acid. So you eat them, right? You eat something that has Shigella, or you eat something that has Salmonella on it. Okay, the Salmonella is more likely to die in the stomach from the stomach acid, so it's going to take more of them to make you sick. Whereas the Shigella, it survives. It survives. Okay, now there's this number that they use, which is ID50. So this is infectious dose 50. So this is like a theoretical number that if they tested, like they did a study, this would be, the ID50 is the number of cells that it takes to infect 50% of the population. Okay, now the stages and the progression of infectious disease. Okay, so this kind of got messed up. It's not lined up right, but so the first stage is the incubation period. So the incubation period, you've been exposed to the pathogen, and you're kind of waiting to see if you're going to get sick, if you're going to get infected. Okay, so you've been exposed to it. So if you guys think about like coronavirus or COVID-19, SARS-CoV. Okay, if you think about in the beginning, right, we started out saying, oh, if you get exposed, these are all the different things that you're going to have to do, and this is how long you're going to have to do them. based on what we were assuming the incubation period was. Okay, now prodromal is, and it's not on the chart in the book, but prodromal is what they call the phase where you think you're getting sick. You start to feel like you might be getting sick. So this is when I'm going, oh no, I have a sore throat. I better drink a bunch of water and I need to take my vitamin C. And I do all these things that probably don't really matter, but it's in my head, right? So I'm thinking, oh, this is where I'm at. Okay, so then illness is where you're going to experience the symptoms, right? And people are going to see the signs. So that's full blown infection. And then convalescence is when you start to feel better and you're recovering. Okay, now in the old days, they used to call like rehab centers convalescent homes. Okay, so now each disease is different, right? So every disease has got a different incubation, different illness duration, different time for convalescence. Okay, now after recovery, or if you get vaccinated, right, memory cells will usually provide protection, and that means this will hopefully get condensed. It won't be as long if you do get sick. Okay, now I put little asterisks here. So asterisk here and an asterisk here, because during incubation or during convalescence, you still could be contagious. Okay, so it depends on the microorganism. Okay, so the pathogen could still be spread during incubation or convalescence. I just thought of something I wanted to say. I can't remember. That's okay. I had the graph. I just kind of make it go like this. So you get exposed, right? Then you have this incubation period, and then prodromal, and then illness, and then convalescence. Right? And at the top, they call this acme, but I don't know that our book does that. But acme is like the peak where you're like, oh, this, if I don't get better, I'm going to have to go to the doctor. I'm going to go to the hospital. So acme is like when they say that fever breaks. Okay, now here is acute, chronic, and latent diseases and how the progression occurs. So acute means the illness is short term, it comes on suddenly. The host is able to eliminate it. Usually the person becomes immune to getting reinfected, but that's not necessarily true. Okay, so incubation, illness, convalescence, bang, 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 happens really fast. Okay, now if something's chronic, the illness is going to last a long time. And that makes sense to you guys. Okay, now if something is latent, if it's latent, you get exposed, you incubate, you get sick, you start to feel better, and it goes into like latency. So it's just hanging out and it's not making you sick. But say something happens, you get all stressed out, or something that triggers it, and you get sick again, you get illness again. So that's latent. Latent, it kind of like just lingers and hangs out, waits for an opportunity to reoccur. Okay, now these vocabulary words are going to describe where the pathogen is. So localized infection means it's just in a specific part of the body. So very specific location. Okay, so if you step on a tack and you get infected, in the hole, that's where it goes. You get strep throat and it stays in your throat. Okay, now systemic means it's going all over the body. Okay, now anytime you see this EMIA on the end of these, it means that it's in the blood. So bacteremia, so it's not necessarily a disease state. It just means that you have bacteria in the blood. you can have some in there and your body's trying to get rid of them and you don't get sick from them. However, if you start to have systemic inflammation because of all the the bacteria start like accumulating in your blood, multiplying, okay, that's what we call sepsis. So septic. Okay, then toxemia, toxins in the blood, and viremia, viruses. Okay, now this is Koch's postulates. So Robert Koch, 1800s, he developed a system or these postulates where we try to figure out what's causing the disease and what the causal agent is. Okay, we've adapted this to modern times, but this is initially how they figured out that like a certain bacteria was causing certain symptoms. Okay, so the first thing is the microorganism is present in every case of the disease. Okay, so we have all these sick mice or dead mice, it looks like. Okay, and if we take a blood sample out, they all have this little bacillus in their blood. Okay, now we want to see if we can grow this and start to identify it. So the microorganism, we have to be able to grow it. We have to be able to identify it in pure culture from the diseased host. And this is where things have modified because technology has changed. Okay, then the next step is the same disease can be produced when we take this pure culture and we put it into a healthy host that's susceptible. So we're going to take that and we're going to put it back in that mouse, and guess what? He died. Now, what we have to do to totally fulfill them is we have to be able to take some blood back out of it and prove that that organism is still in there. That's what caused it. So those are Koch's postulates. You can't do it with humans, right? Nobody wants you to be injecting humans with something to see if they make it sick, if it makes them sick. And there's ways around it because like a virus, you can't grow in a pure culture, but there's ways around it. They've adapted it for modern times, but the concept is still really important. Okay, now here, so this is what I was just trying to say. So limitations of Koch's postulates. So some agents can't be grown in the lab. Okay, so one example is syphilis. So syphilis is caused by a spirochete, and you can't grow it in the lab. Okay, now people that are infected don't always have symptoms. Okay, so people that have cholera or polio, they might be carriers. That's okay. I have to remember, go back. We have to talk about carriers. Okay, some diseases are polymicrobial. So there's more than one microorganism. Okay, so an example of that is lots of diseases in the mouth. It's not just one organism. So chronic periodontitis, okay, so gum disease, right? There's more than one organism in there. So you can't get one organism in pure culture. There are not always suitable animal hosts. So sometimes like a virus or something, you can't take it from a human and put it into an animal. It's not ethical to test the postulates on humans, but in the old days, there are examples of them unethically doing this. Okay, now let's go back here. This is the carrier thing. Okay, so a carrier is someone that can carry the disease and spread it. So so a carrier is someone that can carry the disease. They don't get it, but they can spread it and infect other people. Okay, so the most famous carrier is typhoid Mary. And I'll post a link so you can listen to the story of typhoid Mary. But it's interesting. So she carried typhus, and she was a cook. So I'll post a link to it. It's only probably like 5 minutes long, but they tell the story probably better than I can. Okay, so now mechanisms of pathogenesis. I'm having problems with my Apple Pencil, which is sad. Okay, mechanisms of pathogenesis. So how do they cause disease? Okay, so how do they cause their symptoms? So there's several general patterns. So one is they can produce a toxin that gets ingested. Okay, so you eat the toxin. So the bacteria are growing maybe in food. You eat the food, you ingest the toxin, and the toxin makes you sick. Okay, so two classic examples of food poisoning, Clostridium botulinum, and then, which is botulism, and then Staphylococcus aureus, which is, it causes food poisoning, diarrhea. Okay, so the organisms are growing, they grow in the food, they are producing the toxin while they're living in that, so they're making the toxin. Okay, you eat them, your body actually, the stomach acid kills this, all right, but the toxin makes you sick. Okay, now, some other organisms can colonize the mucous membranes. Okay, so mucous membranes are the openings to our body. Okay, so they get in there, the organisms get in there, they start growing, then they produce toxins. Okay, so examples are cholera, E. coli, this O157H7. This is the one that causes the bloody diarrhea in food poisoning. I can't spell diarrhea. That's not, I know that's not right, but you get the idea. And we'll talk about cholera more. Cholera is not super common as long as there's clean water, but when there's not clean water, it's really bad. So lots of times it's in a refugee camp or places where there are natural disasters. Okay, the other one, which we talked about in chapter 15, is diphtheria. Okay, so the Corynebacterium diphtheriae. Okay, now some organisms can invade host tissues, and they've developed a system to avoid host defenses, and then they start multiplying. Okay, so Mycobacterium tuberculosis. So what's that? TB, right? That's tuberculosis. Yersinia pestis, that's the plague. Not super common anymore. And then Salmonella enterica, which that's diarrhea. Okay, and then some of them invade host tissue and then produce a toxin. So Shigella, dysentery, right? So dysentery is diarrhea. Clostridium tetani. So this is tetanus. Okay, so there's 4 mechanisms, so 4 different ways that they can cause problems. Okay, so now, what makes a good pathogen? And I can't ask you this question because I can't see it, right? But this is one thing to think about. Okay, so what makes a good pathogen? So a good pathogen doesn't kill the host, right? We want the host to survive if you're a pathogen because you want to be able to spread. Okay, so a good pathogen doesn't kill the host. It can replicate without killing. And here I'm just my little plug. There's a video game. I think it's free. I think it's an app, and it's called Plague. And it's fun because you are the pathogen. You're trying to kill everybody else. Okay, now pathogens evolve or change, and the way that they change is they they become more balanced. So they call it balanced pathogenicity. So the more an organism goes through a host and goes from like person to person to person repeatedly, the more it travels through the host, it becomes less virulent. Okay, and the host changes and evolves and becomes less susceptible. So something might start out really deadly, really virulent. So we'll give it like 4 stars for being really virulent. But over time, it's becoming less virulent and the person is becoming tougher. Okay, now, how do bacteria establish infection? So they have to stick. Okay, so they have to adhere. They have to colonize. So that means they have to be able to multiply and grow. And they have to be able to deliver their toxins to the host cells. Okay, so adhering can happen with this protein called an adhesin. Okay, so adhesins are often found on bacteria that have pili. Okay, so they're on the ends of the pili. Okay, so the pili that are covered all over like that, lots of times they call them fimbriae. Okay, they could also be part of a capsule. Okay, or they could be part of the cell wall. And they're going to bind to receptors. So like here's showing how the little pili are going to bind to the receptors on the host cell. Okay, so things that are going to make bacteria more pathogenic, more virulent is capsules, pili. I guess capsules and pili, I didn't say that very well. Okay, now to establish infection, okay, they have to colonize. Okay, so it has to multiply. It has to deal with the host defenses. So it's got to get around the host defenses. Some of them will produce these things called siderophores. And I'm promised my pencil. Oh, siderophores. And the siderophores bind iron. Okay, so, and the iron is going to help the bacteria, but it's going to hurt the host. Okay, the bacteria also have to be good if they're in a mucous membrane. They have to be able to avoid the IgA. Okay, so now the microbe's going to compete with your normal microbiome. So they have to be able to survive the toxins that other microorganisms that are part of you produce. Okay, so they have to be competitive. Okay, now how do bacteria penetrate the skin? There, now it's working again, sorry. Okay, so think about Staph aureus. So how do people get a staph infection? What has to happen? Okay, so usually there has to be a wound, right? So the skin has to be damaged. So any place where you have a hole, it's an opportunity for a pathogen to get in. Okay, so usually it's a wound. So if you scrape your knee up or you get poked with something. Okay, now Yersinia pestis, if you remember what I just said, right? That's what causes the plague. Does anybody remember how the plague gets spread? Okay, so it's spread from fleas, right? So there were lots of rats. The rats had fleas. The fleas would bite the people. Okay, and that would be how the pathogen, the Yersinia pestis, would get spread. Okay, so that would be a wound. Okay, so they can get in through a wound. Okay, they can also get in through a mucous membrane. All right, so that's a hole that we have naturally. Now, if they get in, they have to be able to avoid our immune stuff, right? They need to be able to sneak past our immune system. So they are able to either hide from the host, or they can direct and tell the host cells to take them up. Okay, so here's directed uptake. So the pathogen, which this is kind of crazy if you think about it, it can tell a non-phagocytic cell to engulf them. So they'll release chemicals and they'll kind of like trick them into engulfing them. Okay, so Salmonella does this. So Salmonella is able to signal, right, these cells, and it rearranges the cytoskeleton and it causes these to kind of ruffle, and they fold up around the bacteria, and then the bacteria become part of the host. They get taken in. So kind of crazy. So that's a way that they can get taken in. And the book goes into way more detail about this, and I'm kind of abbreviating it, or I am definitely abbreviating it. Okay, now some of them are able to hide within a host cell. Okay, so they can hide and kind of make themselves look like the host. Okay, now, how do they damage the host? How do they cause symptoms? So there's direct and there's indirect. Okay, so directly means they're making the toxin, and the toxin's going out and it's damaging the tissue. Okay, now indirect means that they're causing an immune response, and the inflammation is damaging the tissue. Okay, now the damage that they cause can help the pathogen exit the body and spread to other hosts. So this is a good pathogen, right? If you can spread to something else without killing the original host, you're a good pathogen. Okay, so an example of this is cholera. Okay, so Vibrio cholerae. So if someone gets this, they have 20 liters of diarrhea a day. Okay, so 20 liters. That's a lot of diarrhea. So the Vibrio releases the toxin, and the toxin damages the walls of the intestine. And the walls of the intestine slough off. They call it rice water stools. So it's the rice stuff is actually intestine. It's like kind of sloughing off, but that's a really good way to spread, right? Things that are respiratory, so Bordetella pertussis, this is whooping cough. Okay, if you cough all over somebody, the pathogen is going to get spread. Okay, now let's talk about exotoxins. Okay, so exotoxins are proteins. Okay, they're going to be secreted, okay, or they get leaked from the cells after the cells get broken apart. Okay, exotoxins can cause, they call them foodborne intoxications. So that's like Staph aureus, where and this is in like potato salad, mayonnaise-y things. Okay, so you get staph, it starts growing in the mayonnaise, it starts releasing its toxins. Okay, and then you ingest it. That's gonna can cause diarrhea, right? Most of the time, toxins are destroyed if something is heated well to the right temperature. Okay, now exotoxins can act locally or they can act systemically. Okay, so they might target just a certain part of your body, or they can be all over. Usually your immune system is going to generate antibodies to the toxin, And if your immune system can't respond fast enough, they're fatal. They can be fatal. So there are 2 vaccines that they're called toxoids. So these are vaccines to the toxins. Okay, and 2 of those are tetanus and diphtheria. Okay, so you're going to make antibodies to the toxins. Okay, so these antibodies are going to protect you from having all of your muscles lock up and you dying, or getting diphtheria and dying. Okay, now if they give someone antitoxin, an antitoxin or like anti-venom, it's basically they're giving someone antibodies. So you're not making them, but we're giving them to you. You're not going to have time to make them. You're going to die, right? So we give them to you to help take the toxin out. Okay, more exotoxins. So exotoxins are grouped into 3 main types based on the tissue that they affect. Okay, so one is a neurotoxin, one is entero, and one is cyto. Okay, so nervous system, entero, GI, right? And cyto, they're going to be more localized. They're different. It's kind of a catch-all. Okay, now they can also be categorized by their structure and how they work. Okay, so there are A, B toxins, there are ones that damage membranes, and then there are these other ones they call super antigens. This is the easiest way to characterize them. Okay, now A, B toxins, basically the toxin has 2 parts. Okay, so if you look at this, the A is the active part, that's the toxin. And then the B subunit is just where it's going to bind. Okay, so basically what happens is the B part is going to bind to a receptor on a cell. So if they're going to affect nervous tissue, they're going to bind neurons, right? And then we're going to be able to take up the toxin. The toxin is then in the cells and can cause damage. Okay, so things that are A, B are cholera, botulism, and tetanus. Okay, so botulism toxin, that's Botox. Okay, now membrane damaging toxins. Okay, they're going to disrupt the plasma membranes. So hemolysins, or it's probably said hemolysin is probably the proper way, but hemolysin is going to make more sense, right? So hemo is blood, and lysin is going to lyse. Okay, so they're going to break apart red blood cells. There are some that make holes in things. So like Streptococcus pyogenes, which causes strep throat, put holes in the cells. And then there are some, so this is called phospholipase. Okay, so remember ACEs are enzymes, right? So these break down phospholipids. So Clostridium perfringens causes gas gangrene. So basically, the toxin in it is breaking down the fats in the cells. Okay, now super antigens, the way that they work is that they are going to overstimulate the T cells, the helper T cells, so the generals, right? They're going to overstimulate that, and that causes what they call a cytokine storm. So it just causes a lot of inflammation. And if the inflammation is like through the roof bad, okay, that's gonna, it can kill you. Okay, so Staph aureus, strep pyogenes can cause it. TSS is toxic shock syndrome. So toxic shock syndrome, you guys probably maybe have heard of it from tampons, right? So in, I guess it was the 70s or the early 80s, they made extra high absorbency tampons, and people would put them in and leave them in for a long time, and bacteria would start to grow on them. And these bacteria were producing toxins, right? And super antigens, which were causing them to go into shock. So not good. So fever, vomiting, diarrhea, in this case, death. Okay, now there are other toxic proteins that are interesting, and we'll talk about them as we get to specific diseases. So there's one called exfoliatin. So it exfoliates. Okay, so Staphylococcus aureus produces it. They call it scalded skin syndrome. It happens in babies. And basically what it does is it breaks down the material that holds the layers of the skin together, and the skin starts sloughing off. Okay, so it's going to spread systemically throughout the body, and their skin just kind of peels off. It's like a bad sunburn, blisters, and skin sloughing. Okay, some bacteria can produce proteases, which are going to break down proteins, lipases, which are going to break down lipids, collagenases to break down collagen. So together, what they'll do is they'll destroy tissue. Okay, and as they destroy tissue, right, the bacteria are going to be able to spread and just keep going. So some like flesh-eating bacteria, that they produce lots of enzymes that do stuff like this. Okay, so I have a chart. Okay, so exotoxins generally come from gram-positive, and endotoxins are found in gram-negative. Okay, endotoxins are found in the cell wall, and exotoxins are found within the cytosol. Okay, so in the cytoplasm. Exotoxins are proteins, and endotoxins, if you think about lipopolysaccharides, okay, so they're lipids and sugars. Exotoxins, yes, they can form a toxoid. No. Heat stable. So, right? I don't know if they're heat stable. I think that these are not heat stable, right? Which makes sense. So if you cook the food, they're not going to cause a problem, but these are more heat stable. Mechanism. So these are going to cause shock, right? And these, let's put this is systemic. This is what it was. Okay, and this can be localized or systemic. So like basically, how does it affect the body? And toxicity, both of them are really can be really bad. I should probably, this is probably bad box to put in here. Yeah, so you don't have to put anything in. It depends on what it is, the situation, how bad it is. Okay, but this is kind of important. So these are coming from the cell walls. Endotoxins are lipopolysaccharides. Exotoxins, these can be secreted. That's probably what I was trying to get at down here. These can be secreted, but the cell has to be broken apart. So the cell has to be lyzed. Okay, now viruses, which we just had a whole chapter on that. So to infect a host, the virus must enter the cell. It uses the host machinery for replication. It has to avoid the immune system. and it can then move to another host. Okay, so the damage that the host experiences can result from the viral infection or from the immune response. And we did a whole chapter on that.